welcome to the first laboratory, Introduction to Medical Bacteriology Laboratory, from the subject MTH67-342 and 342E, Medical Bacteriology and Mycology Laboratory. Here is the learning objectives that you have to learn in this class. So the first one, you have to know the general rules and regulations in medical microbiology laboratory. The second one, you have to explain the principle, method, advantages and disadvantages of the pure culture isolation techniques, which we will learn about the streak plate technique. So you have to know the practice, how to do it, and also photogram staining that we will learn in this class. So you have to know the principle and procedures correctly, and also perform the initial emergency biological spill response procedures. So in the general rules and regulations in the medical microbiology laboratory, the first one, you have to need a personal responsibility that you have to be followed by the rules and instructions in the medical microbiology laboratory. The second one, you need to have the appropriate PPE and wearing it all the time in the lab, such as lab coat, gloves, eyes protection like glasses, the hand hygiene before and before you leaving the laboratory. You need to wash your hands properly with the hand soap and or any detergent. No eating, drinking or smoking in the class. The fifth one is the laboratory practice. The proper laboratory practice has to be performed in the laboratory. So we will work for it. The sixth one, work area maintenance. You have to clean it before and after using any equipment and also counter that you use. Equipment care. Here is if this equipment has been damaged or any malfunction, you need to report to Ajahn or the instructors or any staffs. 8.1 specimen management. This one, you will have so many containers like the bags, like plastic bags, or any container like plastic containers like this. So you need to know which one that you need to use, and also has to be labeled. Okay. The nine, the waste disposal. They are characterized in the infectious waste, child waste, chemical waste, and general waste. So it depends on which trash you need to throw away. Okay. The 10.1 is the accident reporting. All the accidents, spills, exposure, and any injuries, you have to immediately report to your instructors or any staffs. Okay. 11.1 laboratory exit. Do not remove laboratory materials or equipment without permission. And the last one, you have to follow the instructions at all time, or the laboratory supervisor. In case of the emergency happened, the first thing first, you need to stay calm and assess the situation. So after that, you have to alert or notify other people that the accident happens immediately. Okay, so then you can wear the PPE, which consists of like lab coat, gloves, or eyes protection, depends on how big situation is. Okay, after that, you use the absorbent materials that can cover the spill area. Then use the disinfectant and pour it around the accident area. And after that, dispose of waste properly. So you have all of it like absorbent materials, and which is maybe the tissue papers that can absorb the spill. Okay, and then don't use the bare hand or hands that wearing gloves. You need to use the forceps that is inside the spill kits already. So all of the equipment like the absorbent materials or disinfectants is already in the spill kits. Okay, and after that you clear everything. In the end, you have to report this incident to the laboratory instructor. So in this picture show how to wear a lab coat correctly. So first, if you have the lab coat, you have to wear it all the time in the laboratory. The collar have to lie flat and looks neat. The button is, you have to button up all of the buttons of the lab coat. Sleeve have to reach to the wrist level. The lab coat has to be clean and not damaged. The length has to be in the knee length like this. And also in the back, it has to cover all the back completely. Okay, and here is the incorrect one, which maybe roll up the sleeves. This is not allowed. Okay, and not buttons up. Wear the coats that's too long or have the dirty or damaged lab coats like this. And also the flip-flop is not allowed to use in the laboratory. So you need to use the trainers like this, sneakers. Okay, so come to the first lab. What we are going to do in this lab. So we will provide you the mixed bacterial culture in the test tube like this, and also the petri dish of the Escherichia coli, which is the representative of gram-negative bacilli, and Staphylococcus aureus, which is the representative of gram-positive cocci in cluster. What are you gonna do with these? For the mixed bacterial culture, you need to streak it in the blood agar, McConkey agar, and smear it in a glass slide to prepare gram staining. For the E. coli, gram-negative bacilli that's in the petri dish, you need to do the gram stain as well, along with the Staphylococcus aureus. So in the one glass slide, you can smear it all together with the E. coli and the S. aureus here. So manage it to use just only one glass slide. Okay, so after that you can apply the staining at the sink. Before that, you need to don't forget to fix it with the flame. Okay, so here is how to prepare the bacterial smear. Okay, for the procedure, you have to place one or two loop full of the cell suspension on a clean slide. So the glass slide here, you can clean it we're using the alcohol. Allow it to dry, and then after that you use the loop full of the suspension that is in a broad medium. Okay, and then after that you touch it about 1 or 2 times in a glass slide, and then smear it in a circular motion. Spread the suspension into the thin area, which approximately like 1 baht coin. Okay, and then after that allow this smear to air dry, so it takes around I don't know, maybe 10 minutes or more than that. So after it dries, allow it don't forget to flame heat fix the slide before you stain it. And for the solid media, you can use a drop of water, okay, and you can drop it in the edge of the slide first, and use this loop that already flame with the fire that allowed to dry already, and touch it a little bit to have it like a film in the loop, and touch in the middle of the glass slide, and then use another loop to pick up the bacteria inoculum in the disc, okay, and then suspend it in this water, okay, in the circular motion as well as the broad medium. And this is the same procedure, so you allow it to dry and flame with the heat fix. Okay, so in these steps, you will get the mixed bacterial culture one slide, and other slide with the E. coli and S. aureus. Okay, now you can do the gram staining. So the first step, you use the primary stain, which is the crystal violet. Okay, the second step is the iodine as a mordant. The third step is the alcohol, which is a decolorizer. And the fourth step is the safranin oil. So every step, you need to wash the water before other step that will take place. Okay, so for example, if you use the crystal violet for a little bit of time, then after that, before you do the step 2, you need to wash it, rinse it with the water before adding the iodine. Okay. And how to report it? You need to report the color, which is gram positive or gram negative. Gram positive, you see the purple, okay, purple color, and the gram negative one, you see the red. For the shape, if it's round shape, you report as a cocci. If it's a rod shape, you report it as a bacilli. And for the arrangement, if you see the arrangement of the bacteria in the slide, okay, you have to report it as well, such as clusters or chain. Okay, so here is the example of the picture of the gram positive or gram negative bacteria that you can see it. So gram positive cocci in cluster, you can see it as a group like this. This is an in chain, the gram positive cocci. So here is the purple color bacteria, so you report as a gram positive, okay? And it's cocci because it's round and it's in chain arrangement. Okay, something like this. Oh, and also the gram negative, you don't have to report the arrangement, okay? And also the gram positive bacilli as well, you don't have to report the arrangement because you don't see any of the arrangement here. Okay, so here is the report form. So you have to capture your picture under the microscope and fill it here in the tables like this, report it. And also if you have some incorrect, so you have to report it if why it's wrong. Okay. After that, here is the pure plate technique and also the bright plate technique that we will not do in this class. So we will learn in the next topic. And also for the experiment 2 streak plate technique, you're given the blood agar one, and you can see the bacterial growth characteristic on the culture media. You need to identify the number of the distinct colony morphologies present in the blood agar. And for each colony type, you have to record the following characteristics like shape, form, size, age, color, surface consistent of the texture or the texture. For the hemolysis, you have 3 of the characteristics of the hemolysis, 3 types of the hemolysis, which is beta, alpha, and gamma. So the beta one, if that bacteria can lyse the red blood cells, you can see the transparent color like this. And if it's alpha, it means that these bacteria can hemolyze or lyse the red blood cell, but it's incompletely. So it has a little bit of the green color. The gamma one, it means that bacteria cannot lyse the red blood cell, so you can see just only it stays the same like the red color one. Another plate is the McConkie agar. So this one you have to record the growth characteristics of the bacteria on the culture media as well. Identify the number of the distinct colony morphologies represents. And for each colony type, you have to record whether it's a lactose fermenter or non-lactose fermenter. So for the lactose fermenter, if these bacteria that grow in the McConkie agar utilize or use the lactose, these will show you in the pink color. But if these bacteria cannot use lactose, okay, will ferment lactose, it will stay the same like this, like the color quite transparent one or colorless one. Okay, how to report it? So you have to fill your you have to take the picture of the plate, which is a blood agar plate and McConkie agar, to show the bacteria that grow in the agar, and also report the colony on the bacteria on the blood agar, which is maybe 1 type or 2 type. Okay. Here is the biological spill response, that is the experiment 3. In the spill kit, it contains all of these equipments, which are disinfectant wipes, absorbent pads, disposable gloves, protective eyewear, plastic scoop and brush, bags which has a biohazard label on it, paper towel, forceps or tongs, zipper bags, biohazard labels. And here is how to deal with the biological spills, so which I have already explained you. So here you can go to this website to see the how to do it. And this is a reference. If you have any information, if you want to find any more information, you can go to these references. Okay, thank you very much.